Let's do another example using the Hardy-Weinberg equation. We're going to look at a disease called phenylketonuria, or PKU. This is a disorder where individuals who have PKU cannot break down the amino acid phenylalanine, and it can cause a variety of problems for the individual. We're not going to go into that at the moment. We're just going to look at the allele frequencies for this disorder. So PKU occurs in 1 in 10,000 US births. That's about 0.01%. So a very low frequency in the population. So this problem gives you the phenotype frequency of affected individuals. So we are given a phenotype. Remember that I asked you to look at each question, each Hardy-Weinberg problem that you're given, and determine whether or not you are given an allele frequency or a phenotype. So we are told that 1 in 10,000 individuals are affected, which means we are given a phenotype. So the phenotype of the recessive allele corresponds with Q squared from our Hardy-Weinberg equation. So the first thing we need to do is determine the frequency of the PKU allele. So because Q squared is equal to the frequency of affected individuals and also the frequency of homozygous recessive individuals, we can set Q squared equal to 0.0001. That's just 1 divided by 10,000. So to find the frequency of the PKU allele, we need to calculate Q. Q is equal to the square root of Q squared. So Q is equal to 0.1. Now, in order to continue on with our problems, we are also going to need to figure out the frequency of the normal allele, individuals who do not have PKU. So we can do that by using the equation P plus Q equals 1. So P is equal to 1 minus Q. So P is equal to 0.09. So now we have the allele frequency of the normal allele. Now let's calculate the frequency of heterozygous individuals. You might do this because you want to know what percentage of your population are carriers for a particular disease. So to calculate the frequency of heterozygotes, we use the 2pq part of our equation. So 2 times 0.9 times 0.1 is equal to 0.18, which means that 18% of the population are carriers for PKU. Recall that the whole point of using the Hardy-Weinberg equation is to determine whether or not a population is in Hardy-Weinberg equilibrium. So to determine whether or not a population is changing. So we can use this idea of P plus Q equals 1 to help us determine whether or not the allele frequencies are changing. Because if one of these goes up, the other has to go down, and therefore the population has changed. So let's look at the assumptions as they're related to PKU and see if we can evaluate whether or not this allele for PKU is changing in the population. So the 5 Hardy-Weinberg assumptions, let's start with no mutation. So if the population is not changing, we assume that no mutations are happening. So we can sort of assume that mutation rate for the PKU gene is low. The reason is that mutation rates are low in general, and then the chance of a mutation happening within the PKU gene is even lower than that. And so although we absolutely could have a mutation that comes up in the PKU gene that leads to the PKU disease, this one is probably the least often violated because mutation rates tend to be low. So at any single point in the population, we are very unlikely to have a mutation in the PKU gene. And then on top of that, because our population size worldwide is so large, that having one individual with a mutation is actually probably not going to affect our allele frequencies. So we can assume that there are no mutations. We can also assume that there's no differential survival or no natural selection. This is because dietary restrictions control the disease. So individuals who are on this, or have this disease are on a special diet that makes them no less fit than anybody else. No genetic drift. We can assume this because the worldwide population, or even just the US population, we actually use the US data. Even the US data is sufficiently large so that genetic drift is not going to have a large effect on this population, meaning that the allele frequencies for PKU are not going to change by chance because the population is so large. We can assume that there's no gene flow because gene frequencies for PKU in other populations, other countries, are similar to the United States. So even if there is gene flow, the rate of people with PKU coming in should be the same rate as people with PKU in the United States. So they should be moving and flowing at the same frequency. And then we can assume that there is random mating because people are not basing their mating choice on whether an individual is susceptible to PKU. I highly doubt that that is on any of your lists for choosing a mate. And then we can also assume that because inbreeding is not common in the United States, we are not getting preferential mating within family lines. So because of all this, we can assume that the PKU allele is not under evolutionary pressures of any sort. The population is not changing, and therefore the PKU allele is in Hardy-Weinberg equilibrium. For most of the remaining parts of the chapter, we are going to be focusing on talking about the Hardy-Weinberg assumptions in a little bit more detail. This diagram shows 4 of the 5 Hardy-Weinberg assumptions and their effects on allele frequencies. I'm going to very briefly describe them. You should be familiar with all of them, and we're going to go into all of them in a bit more detail and do some examples. So the first one is selection, or differential reproductive success. This is where some individuals leave more offspring than others. This is usually because one of the alleles has an advantage of some sort in the environment compared to the other. And so individuals with that allele tend to survive better, survive longer, and therefore produce more offspring than those who do not have that allele. So our example here, population in generation 1, shows the population undergoing selection. So we see sort of a shift in allele frequencies. It's sort of hard to see what's actually happening here. If you look closely, the blue allele is becoming slightly more frequent within this population. It's being selected for. We can also change allele frequencies by acquiring a mutation. So here we've got a mutation in this population so that now we have a third allele. If the mutation is detrimental and that individual dies before reproduction, it's not going to be passed on. But if for some reason, or just if the mutation is not detrimental, that individual survives, they're going to pass on that allele. And then depending on the other factors, selection, drift, things like that, that allele will change in frequency. It might leave the population, it might increase in population. And if we now have 3 alleles, that of course is going to reduce the frequency of the other 2. So our blue is going to decrease, our yellow is going to decrease, and our red is going to increase. You know, we can also have mutations between blue and yellow, which will also affect allele frequencies. Then we've got migration. So this can be immigration or emigration. So coming in or going out. That is going to affect the population by, depending on the allele frequencies in the other population. So if one of these alleles is really high in a neighboring population and those individuals come in, it's going to increase the allele, that, say, the blue allele in one population, and therefore decrease the yellow. Finally, we have genetic drift, which is where small population sizes result in random changes. This is because the chances of changing the allele frequencies is greater when you have a small number. If you have a large population, you're less likely to experience chance events. The last one is assortative or non-random mating. Now this one is talked about separately because all of the other four change allele frequencies and genotype frequencies. But when you have assortative mating or non-random mating, it doesn't actually change the allele frequencies, it only changes the genotype frequencies. And then after one generation of random mating, you restore the predicted Hardy-Weinberg frequencies. So there are two different types of non-random mating. The first is negative assortative mating. So assortative mating just means preferential mating. You're choosing an individual based on a particular trait. So there are two types. There's opposites attract, so negative assortative mating, or like prefers like, positive assortative mating. In negative assortative mating, individuals are choosing those other individuals with opposite traits of their own. So your homozygous individuals with one phenotype are choosing their mates because they are homozygous for the opposite phenotype. Now, when they mate, they are going to produce heterozygous individuals. So negative assortative mating produces an increase in heterozygous individuals. Now, this tends to keep the population more similar because every time you choose opposite traits, you're just producing heterozygotes again. So you're continually producing individuals that have both alleles. So if we look here at our heterozygous individuals, we don't change the frequency of heterozygotes very much. We do change it, but not as much as we're going to change the heterozygous frequency with positive assortative mating. But if you calculate the allele frequencies, you'll notice that they don't change because no alleles are being lost, selected against, lost due to drift, or increasing because of selection or because of genetic drift. The allele numbers are not changing. In positive assortative mating, individuals who have one phenotype are choosing individuals who have the same phenotype. So that can be homozygous choosing homozygous or homozygous choosing heterozygous. Or we get homozygous recessive choosing homozygous recessive, in which case we are going to increase the number of homozygous individuals and we are going to decrease the number of heterozygous individuals. So you can see the difference in the heterozygotes there. But again, nobody is missing out on breeding because of their alleles. There's no selection working. And so there's no change in our allele frequencies. So non-random mating is not actually changing the allele frequencies in the population. So we're not really seeing evolution. We're seeing a change in the genetic makeup of the population, but the alleles are not changing relative to one another. There is, though, an extreme type of positive assortative mating, which is inbreeding. So inbreeding is where you mate with somebody who is genetically related to you. So your alleles, your haplotypes are identical by descent from a common ancestor. That means that you have the potential, if you are an inbred individual, you have the potential of acquiring a mutation from both sides because each parent is getting it from the same common ancestor. So if we have a family where a mutation comes about that is passed down to their children, the potential for it combining into an inbred individual is higher than if you were just to be a carrier within the population. So we see this individual down here has acquired both mutations from the same ancestor. This grandparent passed it to father and daughter, who were brother and sister, and then passed it down to each to their children. So this is a first cousin marriage, and this is what we mean by identical by descent. So the homozygous individual comes about because a homozygous recessive allele was passed down both sides of the family tree. So the correct term for inbreeding is consanguinity. Consanguinity. It means common blood. So it's just indicating that you have the same blood coming from both parents. So any two individuals from a common ancestor are going to be related by their genetic material. And the chance that somebody is identical by descent, meaning that they have the same genetic information because of a common ancestor, is going to sort of decrease with the number of generations that you are from somebody. So we can determine how likely it is that someone is identical by descent by calculating the coefficient of relationship, which is just a measure of relatedness between two individuals. And like I said, the chance that two individuals share the same genetic material at a particular haplotype or allele. And this is due to a common ancestor. So the coefficient of relationship, or how related two individuals are, is just equal to 1/2 raised to the n power, where n is the number of individuals the number of links or steps between two individuals. So these two individuals, C and A, are connected by 4 links or 4 generations. 1, 2, 3, 4. So in this case, their coefficient of relationship is 2 to the 4, or 1/16. That means there's a 1 in 16 chance that they have an identical haplotype or allele because of this common ancestor. So if we have an inbreeding situation and our first cousins from the previous slide have a child, this individual has an increased chance of sharing having the same genetic information from a common ancestor. So this might be if this individual passes a little a all the way down on both sides, and this individual becomes little a. Now, if this is a homozygous recessive disorder, this is going to be a problem for that individual. inbreeding is not only a problem for people, it is a problem for populations in general. And populations can get into what ecologists call an inbreeding or extinction vortex. So if we think about the other Hardy-Weinberg assumptions, one of them is genetic drift. This is where chance events can change allele frequencies in unexpected ways. And remember, we said that if it's a small population, that those chance events are going to have a greater effect than in a large population. When you have a population that is inbreeding, generally, if you're talking about organisms, it's forced inbreeding because their population is small and they have no alternatives but to breed with close relatives. You end up increasing the chances of those homozygous recessive detrimental alleles. And so if you have a population that is subject to inbreeding because they're already small, because there are no other options, and then random genetic drift changes your allele frequencies, you're going to have a loss of genetic variability. You're also going to have a reduction in fitness because those deleterious alleles are going to end up increasing in the population because they're not being bred out or being kept low by large population size. So because of this loss of genetic variability, you're going to have a reduction in individual fitness and the ability of the population to adapt. So they are going to have lower reproduction, they're going to be less fit, so they're going to have higher mortality, and it's going to decrease the population size even more. And this can continue and continue until you're left with no individuals in the population. So once your population size gets real small, you begin to accumulate a lot of problems in that population. Inbreeding, genetic drift, coupled by higher mortality, just continues to decrease the population size. So let's calculate our coefficient of inbreeding. This is similar to our coefficient of relationship. So we are going to be calculating the degree of relatedness due to inbreeding. So basically the chances that they are identical by descent from the same common ancestor. Now remember, this is a problem because any deleterious alleles are going to get concentrated by that inbreeding and increase this individual's chances. So the very first example, we had this example. I'm going to redraw the letters here. Let's say that this individual has a child and this individual has a child. Now, we calculated the chances that these two individuals were going to be identical by descent. And if we add their children into the link, there are 1, 2, 3, 4, 5, 6, 1/2 to the 6th. That's the coefficient of relationship of these two children. Now, what if instead of A and E having children outside of this family, they have a child together? I drew them both as females. That's not going to work. You know what I mean. All right. So if instead of breeding outside of their family, they have a child within, their coefficient of inbreeding or coefficient of relationship due to inbreeding is actually going to be slightly higher. So the chance of these individuals having a child with a deleterious allele that is due to the common ancestor is going to be higher than this individual or this group. So if these individuals have a child outside, they have a 1.6% chance of their children being identical by descent. But if they have a child together, they have a 3.2% chance of their children being identical by descent. So you can see that there is a basically twice the chance of this individual inheriting a deleterious allele. And actually, if individuals are too closely related, they are going to have, you can see the effects almost immediately. So if brothers and sisters have children together, you see the immediate effects of inbreeding. Now, the reason is because they estimate that everybody is heterozygous for at least one deleterious allele. And so if your sibling or your first cousin also is a carrier for the deleterious allele, there's a chance that you're going to pass it on to the next generation. So just to summarize, your coefficient of inbreeding is going to be 1/2 raised to the n, where n equals the number of individuals that are shared. You can also still consider n as the number of links. So just like the coefficient of relationship, we've got 1, 2, 3, 4, 5 links, or we have 1, 2, 3, 4, 5 individuals link them. You can do it either way. Now, what if there are multiple common ancestors? So this shows half siblings, actually. This is mother with 2 different fathers, so B and D are half siblings. But if you were to have a situation where you have full siblings instead, there are actually 2 different roots for this individual to be identical by descent. And that is the root between the child and the mother and the child and the father. So if we draw, let me get a different color here. So if you have 2 common ancestors, you have to use the sum rule, because you can be identical by descent by 2 different paths. So you can have path 1, 2, 3, 4, 5, 6. Sorry, that should still be 5. 1, 2, 3, 4, 5. Oh, because here, 6 that way. Sorry. Let's draw it this way. All right, so 1, 2, 3, 4, 5. So 2n to the 5 through mom, plus through dad. 1, 2, 3, 4, 5, plus 1/2 to the 5 through dad. I'll do some more examples of this in another video that is just examples. So how much inbreeding or how much relatedness is too much? Well, it turns out that somewhere between 5 and 10% coefficient of inbreeding is sort of the magic number. Once you get down below 5%, you don't see the problems that you would normally see with inbreeding. You see less chance of those deleterious alleles accumulating in an individual. This is why in Western culture, it is, and specifically in the United States, it is illegal to marry somebody that is more closely related than second cousins. This is because if you look at first cousins, the coefficient of inbreeding is actually 6.25%. And then once you get out to second cousins or even half first cousins, which means you're related by somebody with a different parent, you get down below that 5%. So there's less chance of those deleterious alleles accumulating in a particular individual. Now you can see these effects of inbreeding rather quickly. And if you look at Animals like some specific purebred dogs, you actually can see problems because of their inbreeding. They've been selected for and bred within their particular family lines or close family lines for so long that there are problems associated with some of the traits, some of the inbreeding that happens to come up. So just keep in mind, less than about 5% is sort of your magic number. I mean, not that most of you are probably worried about this, but if we're talking strictly from a genetic standpoint, 5% is the magic number. You want to keep that coefficient of inbreeding lower than 5% if you want to keep your population healthy.